So this is topic 8, so cell part 1. Cells all have boundaries. So boundaries like membranes, blueprint of information DNA, ability to harness in energy and material from the environment. They're all self-replicating entities, smallest unit of life, cell coming from pre-existing cells, and delimited by plasma membrane, which is the same thing as boundaries, containing DNA, which is the same thing as blueprint and information of DNA. Cells must synthesize proteins and specific functions that each protein has, capable of reproduction, copying DNA, replication, and divisions. So talking about cells, there are two types of cells, prokaryotic and eukaryotic. Let's first talk about prokaryotic. Prokaryotic cells don't actually start talking with eukaryotic. Eukaryotic basically have true nucleus. They all have nucleus, and they are membrane bound. So they have nucleus and organelles that are membrane bound. So they have membranes, and they are typically larger, 10 to 20 micrometer. And prokaryotes are before nucleus, so there's no nucleus. Instead of a nucleus, they have a nucleoid, which is like a space, open space with lots of things in it. No membrane bound organelles. There's no membrane bound nucleus. There's no nucleus in the first place. But they're really small, 1 to 2 micromillimeter, and they are really good for fusion because they are small. So compared to their volume, their area is a lot. So there's the area to volume ratio is a lot bigger in smaller cells than bigger ones, because obviously if it's small they have more surface area ratio compared to what their volume of the entire body is. So it is actually more beneficial because they can do more fusion and therefore more surface area for contact, for reaction, for interaction with the environment. They are asexual reproduction, so they cannot I actually don't know what is asexual. Okay, so they're able to reproduce by themselves, like splitting up into two. Just they're single parent producers. They may or may not need oxygen, so aerobes, anaerobes, and facultative anaerobes. So aerobes need, don't need, and these are both is fine. So they can survive without it, but they can survive with oxygen as well. So more surface, more contact, and they are double-stranded genes. So they also have double-stranded genes. They also have DNA. Yeah, if it's double, yeah, they do have DNA, and the double-stranded just kind of implies DNA, but they have DNA, and of course they also have RNA. Yes, so they have RNA and DNA as well. Moving on, in the prokaryotic cells, there's this huge bundle of stuffs in it, and that's the circular large chromosome, and there's protein in there too. And the circular large chromosome is just a really huge ring like this of chromosome, but it's mixed up in here like this. It looks like a bundle of cotton, and there is ribosomes. So these little ones are ribosomes, and I also need to draw other stuff, which is more little tiny dots, and these little tiny dots are plasmids. So they're circular extra chromosomal DNA. They're non-essential genes, and these are for example, antibiotic resistant genes. At least I think it is genes, and these can pass on horizontally. So these are genes that can be passed on, not hereditary. So that not generational, but more horizontally. So from one prokaryotic cell to another prokaryotic cell, like the gene can move from one cell to another, literally not their offspring, just another one. And they're small plasmids, they're small, there's a lot. These are DNA molecules that turn into mRNA, and that can also synthesize proteins. So plasmids can make proteins. They're little ones here. They can turn into mRNA, make proteins. And they have cell walls, so they are there's a lot of strengths because of the cell walls, because they can protect, stabilize, support, and protect them against lysis. So lysis is basically exploding because of the interior is less concentrated than the exterior, then the substance inside here will want to get out, and that will cause it to expand and explode, which is called lysis. And because of osmotic pressure, the lysis can happen due to osmotic pressure. So these cell wall will prevent them from having too much lysis. Bacterial domain, they are composed of peptidoglycan. So the cell wall for bacteria are peptidoglycan, but the domain in archaea, their cell walls are composed of other stuff, not peptidoglycan. It can vary. And yeah, so nucleoid are for archaea and bacteria. So archaea and bacteria both don't have membrane-bound organelles or nucleus, so they are prokaryotes. And they have ribosomes, like I say, they have plasma membrane, they have villi, they also have flagella, which is this very long, well, not very long, they have many flagellums. They're long appendage for movement. They will just be able to move. Moving on, we have eukaryotic cells. So eukaryotic cells have nucleus, unlike prokaryotic cells. They also have ribosomes, but they also have other membrane-bound organelles, such as, well, we want to be talking about a lot of those. They both have phospholipid bilayers. Both eukaryotic and prokaryotic cells have phospholipid bilayers for their membranes. So prokaryotic cells are not double membraned, and neither is animal cells. So the double membrane is only for plant cells because they have a cell wall and a cell membrane. Okay, I'll leave a note for that. Just ask her later. So let's talk about, so there's a lot of organelles in here, and these are all called endomembrane system. Endomembrane is everything inside the membrane. They are structured and divided into multiple internal components. Organelles are not distinct, so they all communicate within to each other by membranes of bridges or by vesicle budding, small membranes enclosed sacs. So vesicles are little sacs of information or other substance or other signals. It could be nutrients, etc. And they're going to be transported within the organelle. So they're all not like individual distinct. They're all communicating with each other, transporting vesicles, yeah, working together. And we have nuclear envelope, which is the envelope of the nucleus, or nuclear membrane, you could say. Endoplasmic reticulum, there's soft and rough. Golgi apparatus, plasma membrane, vesicles. So let's first talk about ribosomes. Did I talk about ribosome here? Oh. Okay, so let's talk about ribosomes. It's one of the organelles. Yeah, so it's just one of the organelles. So ribosomes, they are protein synthesizers. So they'll synthesize proteins. They will make RNA sequences into amino acids into polypeptides. Translate RNA into polypeptides. So into amino acid chains and then to polypeptide. Polypeptides are just proteins. Okay, so polypeptides are the linear chains of amino acids linked by peptide bonds. Polypeptides are linear chains of amino acids. but they're not necessary. Not all polypeptides are proteins, but we'll look into that later maybe. Now we have two types of ribosomes: free ribosome and bound ribosome. Free ribosomes are the one floating in the cytosol. Cytosol is the soup. Inside there, just the liquid portion is in the interior of the cell, and these synthesize proteins that do not leave the cell. So the ribosomes synthesize proteins. Free ribosomes are for the proteins that do not leave the cell. Bound ribosomes are for the proteins that do leave the cell, and there are membrane proteins, so they synthesize membrane proteins, proteins that are on the membrane or that leave the cell. Yeah, and these are attached to the rough ER. There's a lot of bound ribosomes on the rough ER, and rough ER, which is what we're going to talk about, are primarily things that synthesize protein again, but to be secreted out of the cell or embedded in the cell membrane. So this is endoplasmic reticulum ER. They are attached to nuclear envelope. More than half of the total membrane is in the eukaryotic cell is the endoplasmic reticulum, so it's one of very big organelle. They are forms of tubules and cisternae network. Basically, their shapes, tubules are like tubes, cisternaes are just these wave-like skins. And rough ER, they are proteins. They form proteins, they synthesize proteins. And ribosomes on the surface will synthesize these proteins. And they are connected to organelles such as nucleus. Their function is to synthesize the secreted out protein, like I said, and hormonal production. So hormone production, they will create proteins and create hormones. Enclosed. So they also pack products in the transport vesicle to deliver to the Golgi apparatus. So they will pack these products into the transport vesicles, like the vesicles that are bubbles, to go to the Golgi apparatus. And now let's talk about the smooth ones. So the smooth ones are, there's no ribosomes on them, they're smooth. They are primarily for synthesizing lipids, but also mostly lipids. So synthesize triglyceride, phospholipids for the membrane, cholesterol for the membrane fluidity, and steroid hormones that are other functional signaling hormones. And these are, like I said, all from membranes. They make triglyceride, which I said right here, which is one of the most abundant, most main body fat, and lipid synthesis for phospholipid steroids, I said, or glycogen. Yeah, so we'll talk more about glycogen for now. They also do a hydrolysis of polysaccharides. So smooth ER, lipid synthesis, polysaccharide hydrolysis, detoxification of drugs, detoxifying. So smooth ER equals detoxifying. That's pretty important. Store calcium ions. This is important for the muscles. They will regulate Ca2 plus calcium ions. And by hydrolysis of polysaccharides, we mean glycogen. So glycogen are either broken down or formed, depending on if it's high blood sugar or low blood sugar. It's to maintain the metabolism of carbohydrates. So these sugars. I think we call this homeostasis. Yeah, so liver are able to liver have a lot of ER and have a lot of smooth ER. They also have a lot of ribosomes and other things, but ER a lot because they need to regulate a lot and they need to detoxify a lot of substance in the body, and especially sugar to regulate the blood sugar level. And ER is also needed in the protein needed in muscle proteins in muscles, because Ca2 plus ion signals are required for muscles contraction to be triggered. So the muscle contractions are triggered by these ions. calcium ions. Lots of smooth ER in the muscle and the liver because of detoxification, lipid metabolism, carbohydrate metabolism, so fat and sugar metabolism, detoxification, help regulate blood sugar. Now let's talk about Golgi. So Golgi is where all the things from the rough ER go into. So the rough ER will make vesicles packed into a Golgi, and rough ER they are doing where is it? Rough ER they are mainly synthesizing proteins, and I think they also synthesize some sugar. Okay, they're just mainly proteins. Mainly proteins that are secreted out or embedded in the cell membrane. So some of them are embedded in the cell membrane, and most of them are secreted out, closed. So they make these products to go to Golgi. So now they come to Golgi. They're not continuous like ER. With other membranes, other organelles. There's a cis phase and a trans phase. Cis is the forming phase, trans is the mature phase. So go in from the cis phase and get out from the trans phase. And what is being done is packaging. They do modification, sort, packaging, proteins and lipids and enzymes and hormones. I think they do hormones. Yeah, hormones. They modify and pack hormones and enzymes. I should add hormones. So all those, they're gonna be packing it for them to be delivered across organelles. So go to be going everywhere in the cell afterwards. Synthesizing polysaccharides, synthesizing sugars, modifying, packing. So this synthesizing is interesting because we kind of know this already. It's glycoprotein and glycolipid stuff that we talk about in the previous topics. So carbons are added or taken away from proteins. So that's the adding, removing carbohydrate to the protein that we're talking about here. And these are done to proteins and lipids to make glycoprotein, glycolipids. Those are both signaling signaling molecules that are sticking out of the cell to do cell recognition and signaling other enzymes out there. And to renew, modify plasma membrane. This is very important for the membrane because these secreting out proteins, not proteins, but yeah, over here. The smooth ones will make lipid synthesis for the phospholipid and the membrane lipids. There are lots of membrane lipids that are formed here to go to the membrane to renew or to modify the plasma membrane after going past Golgi. So either way, ER material will go past Golgi to be packed and actually delivered. So it's like a post-mail place. Yeah, so they add sugar to become glycoprotein, glycolipid. And now these guys, Golgis, okay, they are able to synthesize sugar and modify proteins and hormones and enzymes and stuff. They are released from cells, by the way, they're released from cells. They're getting out of the cells. Secretion is all about getting out of cell. But they also synthesize lysosomes. They produce lysosomes. And when so producing lysosome is all these little organelles, I think they're called organelles, inside a really acidic pH of 5, containing lots of enzymes, so lots of type of different enzymes. Enzyme containing vesicles made by Golgi apparatus, like I said, membrane bound proton pump, some sort of proton pumps that makes it very acidic, optimal for degrading enzymes. Degrading enzymes, what does that mean? I don't know if I'll be able to come back to this later. I'll put next to it. And these are for breaking down bacteria, other stuff that we don't need, regulating damaged organelles, and they can also do autolysis, so self-breaking, so self-killing. suicidal a bit. They're gonna break down, so open up all the lysosomes in their cell so that the pH5 is gonna get out, and all the enzymes are gonna die, I think. Yeah, I'm not sure. I think it's the enzyme that are dying. Yeah, degrading enzyme means the enzymes are dying. Because enzyme, they will stop functioning. They will malfunction from pH of 5 because they are proteins. And some pH of 5 is really bad for their well, they will denature at pH 5 usually. So now we have lysosomes. Lysosomes break down these guys, okay? So peroxisomes are other individual organelles that are also a lot found in liver. So liver has a lot of these organelles actually, like many of them are in liver. So peroxisomes are for detoxifying. Peroxisome are for detoxifying. They're detox machine basically, and present in all cells, but a lot in metabolic active cells, metabolic active like liver, who are able to detox a lot. And their function is to protect the cell from potentially damaging effect of free radicals during catabolism. So catabolism is a breaking down. When there is a breakdown of fatty acids or other organic compound like alcohol, there is production of peroxide, and peroxides are really dangerous for they're harmful, so they need to be absorbed. So what peroxisomes do is to first break them down and absorb these toxic peroxide stuff, both generate and eliminate peroxide. And catalase enzyme, for example, is something that is it's type of enzyme that can break down the peroxide into oxygen and water. At least I think. Okay, let's now talk about mitochondria. Mitochondria are double bilayer. It's one of the two in the cell that is in the animal cell. There's double bilayer. One of them is the nucleus. It has double membrane, and the mitochondria also has double membrane. And this is one of the cells that had gone through endosymbiosis, coming from another cell. So it was an individual cell on its own, was eaten by a eukaryotic animal cell, and now it just kind of lives together and works together. So mitochondria is a powerhouse machine. ATP powerhouse, and especially cellular respiration powerhouse. They contain lots of they contain ribosomes, because they themselves were individual cells, so ribosomes are kind of everywhere in all the cells. Are circular mtDNA present, so they have mtDNA, mitochondrial DNA in there, since they were individual cells before. Double bilayer protein, closed lipid, protein, filled with enzyme. They also have enzymes in there. What is that? And they're smooth in the outer membrane. Inner membrane has numerous folds called cristae, which are these guys, these folds. And these folds are useful because they can do lots of functioning, like reactions for cellular respiration and ATP production. And these are called cristae. And there's enzymes on these folds. They have a lot of dots on it. It's called a cristae surround fluid content matrix. So it's like a matrix inside here. There's lots of things going on. And function is to take chemical energy from food, like glucose, turn them into ATP to make molecular energy. And like I said, there was a bilayer. So there's the inner membrane, which divides the mitochondria into the inner membrane space, so between these spaces, narrow space in between, and the mitochondrial matrix, which is everything inside. And like I said, there's enzymes, there are mitochondrial DNA, mtDNA, ribosomes, fluid. So that's the matrix, is just the fluid inside mitochondria. Cellular respiration in the inner membrane. Oh, okay. So cellular respiration is done in this thin place, I'm assuming. Make sure that later on. And they contain ribosome, mitochondrial DNA, enzymes. So now let's talk about the endosymbiosis. They are eaten, so they have DNA and ribosomes, all of these endosymbiotic cells, and they're originally prokaryotic cells. And they're engulfed. The word engulfed means just being eaten by the eukaryotic cell, and they're the whole cell engulfing these little cells. And now they evolved to a single cell. So that's kind of something that explains how eukaryotic cells are not like really distinct. Eukaryotes are not from single lineage, they are hybrid of like archaea and bacteria, ancestry of fusions of events, and there's just lots of archaea and bacteria history or branch roots, and that's also because of these symbiotic theory that eukaryotic cells are actually host cells that engulfed other prokaryotic cells such as the mitochondria and the plant cell. No, the chloroplast, which I didn't talk about. So chloroplast is the fruits, not the fruit, plant cell component organism. They were also engulfed by plant cell. Well, that was the host cell. And these are able to turn energy of the sun into sugar, synthesize sugar. And that's called photosynthesis, obviously. Double membrane, inner membrane, outer, inner membrane bound components. One of the membrane inside the chloroplasts that are containing chlorophyll is called the thylakoid. So thylakoid are the membranes of chloroplasts containing chloroplast in there. They're able to absorb light, turn it into chemical energy called ATP. So yeah, they're able to make sugar, but they're also able to make ATP. That's a bit confusing. I think it's both. I thought it was only sugar. Now they also have their own DNA, their own ribosome inside their own matrix. So anything that's inside a big bundle is called matrix, apparently. Okay, I think we went over everything. And that's it for